Hello and welcome to my poster. My name is Yang Pan. I am a PhD student graduated from UCLA. I'm currently working at Children's Hospital of Philadelphia. So the title of my poster is PEGASAS, a pathway-guided approach for analyzing pre-mRNA alternate splicing during cancer progression. I also have a talk at ISMB this year, so if you are interested in this work, please come and join my talk. Since I have this full-length talk and also a paper published, the goal of this poster is just to give you an idea of what is PEGASAS. So starting from the first column, so the specific question we're trying to answer by developing PEGASAS is that if you're familiar with RNA-seq analysis, especially alternate splicing analysis, you may be aware of that there are actually plenty of data sets and there are large-scale tools that allow you to identify massive amount of splicing events in this dataset. For example, a recent pan-cancer analysis have identified hundreds of thousands of splicing alterations in tumor transcriptome. So basically, the capability of identify alternate splicing is not a problem anymore. But what remains challenging is that the interpretation of those hundreds of thousands splicing events, especially how they are being regulated during tumor progression. So over the past decade, people studying cancer genomics has established oncogenic signatures to reflect the tumor progression and oncogenic activation. So without that, to leverage those oncogenic signal signatures, we should be able to capture alternate splicing events that are associated with tumor progression. And this is the idea behind the development of PEGASAS. So here I show you the QR code for both the GitHub repo and also the web server. So if you're interested in the tool, please check it out. And below, this is a diagram of the framework of PEGASAS. Without getting into the details, I just want to show you that PEGASAS can start by taking RNA-seq data from patients across different disease stage. And then it branches into two calculations using the same RNA-seq data. And these two calculations are actually orthogonal. They are pathway activity and alternate splicing. And with that, we're able to perform a correlation analysis with permutation. And eventually, we're able to identify oncogenic pathway correlated alternate splicing events. So after we developed the tool and several steps of evaluation, we applied this to study data set we compiled of prostate cancer. It's a very comprehensive data set with nearly 900 samples from different sources of the data. And in the PCA plot in the middle, you can see that samples were able to cluster by their disease stage using the splicing information. And at the bottom in the middle column is basically the PEGASAS result, where we perform the PEGASAS against this data set using different oncogenic signatures. And as a result, we're able to identify a set of splicing alterations that are associated with important oncogenic signatures like E2F, MYC, mTOR, and so on. And what we found really interesting is that if you're looking at the splicing events that are happening in which genes, or the genes that harboring those splicing events, predominantly they are coming from spliceosome-related genes or mRNA binding-related genes, suggesting there is a autoregulative mechanism going on, and the splicing machinery is a major target of oncogenic-driven alternate splicing. Just to give you a concrete example, this splicing factor SRSF3, as I show here, shows a strong anti-correlation between the exon skipping event and the MYC pathway activity score. In a different sashimi visualization, we can see that this middle exon of SRSF3 decreases steadily from a normal prostate all the way to metastatic prostate. And for that, we come to this model here. Because the middle exon contains a premature termination codon, so the inclusion of it will lead to a form that triggers the nonsense-mediated decay, which will eventually lead to the decrease of the protein product. So this is actually autoregulatory mechanism here. And this is actually not new. This has been discovered for more than decades ago, exists in many SR proteins, and also proved to be evolutionarily conserved. And in our data set, with MYC pathway-driven splicing alterations, there are a large proportion of them actually may be involved in this mechanism. And what is new here is that we found that MYC might help those genes by leveraging this splicing event to escape from this autoregulatory mechanism to achieve a dysregulated high-level repression of the protein product. So to further prove this hypothesis generated from large-scale correlation analysis, we performed experimental perturbations where we designed and engineered MYC-dependent cell lines derived from prostate cells to reflect the MYC overexpressed prostate cancer. And we were able to validate a large proportion of those MYC-dependent splicing events. So as a summary, I just want to say that PEGASAS is a big data framework we developed to investigate the interplay between autoimmune splicing and oncogenic signals during the tumor progression. And it is also a very flexible framework to study other types of RNA processing dysregulation. So if you're interested, feel free to contact me or check out my paper and talk. And thank you for your attention.